Welcome to our second fully vodcasted lecture. If you're watching this from the lecture hall, then it is Friday, January the 31st, and probably you're beginning to sort of narrow your focus, your micro focus right now to studying for the exam that is on Tuesday. We've talked a lot about the exam, the format, and the overall protocol for the exam. So I'll see you sometime during the day on Tuesday, February the 4th. And of course, again, do come to the lab to Egg 5030 to take that exam. So wander on into that room and I'll get you set up, or one of the TAs will get you set up with an exam. Remember that I do like you to please be done by 9:30 p.m. So please plan according to that so that you can be done and ready to leave by the time that hour rolls around. There's a review posted online, and that's on the lab web or on the lecture website under additional information, and you'll be able to find that there. Also, we'll have face-to-face reviews, one 12 to 1 p.m. I had briefly mentioned that would be in the classroom, but of course there is a class right after ours, and so we're going to have to come upstairs to the lab. We'll probably be in Agriculture 5028 from 12 to 1 p.m. on Monday, and then from 7:30 to 8:30 p.m. online, we'll have a review on Canvas or on the big blue button feature of conferences there. So either way works totally fine for me, and I'll be excited to see you in any of those, and or to come back and review the recording that'll be available for the online review if none of the reviews work for you. Again, the Gram stain report is due on Thursday, February the 6th, so that will have been assigned in lab yesterday for everyone to begin working on that small but very significant lab report. I'd like to jump back in because, let's face it, this announcement stuff just gets boring. And when we left off last time, we had been talking about gram-negative cell walls. So let's go back and turn our attention to the gram-negative cell wall. And I'm going to draw your attention first to this really cool model that we passed around in lecture. So notice that this is a nice model of the gram-negative cell. And what you can see is the cytoplasmic membrane and then the cell wall. The cell wall is comprised of the periplasmic space and the thin peptidoglycan. Here, this student has chosen to show 3 layers of peptidoglycan. It may be as thin as 1 layer. And then you've got the outer membrane, and remember that the outer membrane has an outer leaflet that contains a lot of LPS. So the student chose to show LPS with the orange head groups and the core polysaccharide and O-specific side chains up there in that region, and the lipid A region down here. So that's the LPS. Those are 3 LPS molecules in a row right there. And then you've also got some other components of the outer membrane, and that's what I want to bring to your attention now. Notice the very large proteins that span the outer membrane. Those are called porin proteins, and porin proteins are have very large pores, as the name might indicate, and they allow molecules between about 600 and 700 daltons to enter into the cell. That is literally, so if you think of sucrose, right, common table sugar, that's got a molecular weight of like 320 something. So that's going to shoot right into a porin protein. But if the molecule's really large, then it's not going to be enter. So we might say that unlike the cytoplasmic membrane that is incredibly exclusive about what can get in, porin proteins allow a lot more in. It's less exclusive, though somewhat exclusive. Now, another molecule that I want to draw your attention to is called lipoprotein, and in the model, the student here is showing that lipoprotein is holding together the outer membrane and the peptidoglycan layer. So that thin peptidoglycan layer is sort of rooted to the outer membrane by what we call the lipoprotein, or more specifically, bronze lipoprotein. So let's look at that and write that down. Porin proteins and lipoproteins joining together the outer membrane and the peptidoglycan, whereas porin proteins, these are playing a role in allowing the movement of molecules from the extracellular space into the periplasm. So things that are of relatively small size can pass in. That is to say, it's not as incredibly exclusive or selective of about what it allows in as the inner membrane is. So that's due to those Porin proteins. But a picture says 1,000 words, and let's take a look at this image where you can see the Porin protein spanning the outer membrane. You can see the LPS molecules in the outer leaflet of the outer membrane. You can also see the bronze lipoproteins knitting together the peptidoglycan and the outer membrane. And you can recognize the periplasmic space there. And you can see the region there where the thin peptidoglycan is found. Remember how we said that there's a lot of enzymes at any given time hanging out in the periplasmic space. They may be molecules or enzymes involved in synthesis of peptidoglycan, maybe they're involved in bronze lipoprotein attachment, or maybe they're things like transport proteins that are involved in bringing molecules to these transporters and getting them into the cytoplasm. So it's a very urban, very busy, kind of big city atmosphere inside of the periplasmic space. So the gram-negative cell wall, very different from the gram-positive cell wall, and hopefully you're starting to get down some of those differences and feeling solid about what differentiates gram-positive from gram-negative, and therefore it should be pretty easy for us to do a little bit of practice. Let's think about a bacterium named Klebsiella pneumoniae. This is not usually a pathogenic bacterium, but it can cause, if someone is immunocompromised, it can cause pneumonia in those people. So we want to know which of the following would typify the cell wall of this bacterium. So notice that it is gram-negative. So perhaps one of the first things to draw your attention to as a part of the question is just that this is a gram-negative bacterium. So we know that it's going to be typified by all of those components that we just talked about. And we can maybe find or select some of those components. But first, let's just think about answer A and whether or not it would have a thick layer of peptidoglycan. Well, remember that that wouldn't be true because it would have just a thin layer. So remember the very thin layer of peptidoglycan there. So we can cross off A. Lipoteichoic acid, that can be crossed off as well. And that's because lipoteichoic acid, remember that that is the polymer that joins or that is bound to the cytoplasmic membrane and extends off of gram-positive cells. So in fact, we can turn this model around and we can find lipoteichoic acid on the gram-positive side. So here we've got the lipoteichoic acid attached to the cytoplasmic membrane and extending from a gram-positive cell wall. Next, lipopolysaccharide, absolutely, right? LPS is that major molecule that constitutes the outer leaflet of the outer membrane of a gram-negative, and like all cells, a negative charge. So the answer that we come up with for this question is F. C and D would both be totally rad answers. I want to make a note that some of you might be craving some conversation. I can hear Thomas right now thinking, how do archaeal cell walls differ from bacterial cell walls? That is, Rachel said that only bacteria have peptidoglycan, but we know that archaea also have cell walls. How do they differ? Well, one note on that is that they don't have N-acetylmuramic acid. They don't have NAM. So they can't make a peptidoglycan molecule that would be equivalent to what we see in bacteria. They have a molecule called pseudomurine that has sort of a somewhat structural similarity, and they can build cell walls. So later when we talk about archaea, towards the end of the semester, we'll do a survey of them, and we'll actually do a virtual field trip to Yellowstone, so that'll be really awesome. And we'll go ahead and talk about more about the structures of the archaeal cell. So you can anticipate that, breathlessly anticipate that, shall we say. I'd like to talk now, though, a little bit about a concept that I know you've been exposed to before. This is osmosis. You know, we have that sort of, I don't know, what is it? It's like the urban use of the term osmosis where we think, yeah, if we sleep on top of our book, maybe it'll come in by osmosis. So while that's a sort of urban dictionary use of osmosis, it is not the technical definition of the term. Instead, what osmosis means is the movement of water. So the movement of water in response to solutes. Water always goes from a concentration of lower solute to higher. That is, when we look at a cell, and on the left, upper left here, we see this, looks like gram-positive bacterial cell, where we see a thick peptidoglycan layer, we see a cytoplasmic membrane, And notice that inside of the cytosol, there's a higher concentration of solute than outside of the cytosol. So water, in this case, is totally going to rush in to this cell. And when it rushes into the cell is going to bulge. And you might be thinking to yourself, the cell is going to burst. But what prevents the cell from actually literally bursting? Remember that rigid peptidoglycan, thick in a gram-positive? It's just going to push the cytoplasmic membrane up against that peptidoglycan. So the cell wall tends to help the cell from bursting as water rushes in. So let's talk terminology here. If the solute concentration outside the cell is lower than inside the cell, we say that the environment is hypotonic. Hypo means less than. So that term, that prefix, hypo, means less than. So there's lower solute concentration outside the cell than inside, so it's a hypotonic environment. We know that that cytoplasmic membrane gets smushed up against the cell wall, and this actually is somewhat enjoyed by bacteria. They actually kind of like it. It gives a little bit of turgor pressure, and they kind of like that just taut membrane up against the cell wall. Now, what if this were mycoplasma? And I think a couple of you raised your hands saying that you had, was that Katie who maybe raised your hand saying that you'd had walking pneumonia? If you've had walking pneumonia, remember that that's caused by mycoplasma and it lacks a cell wall. If mycoplasma had too much of a hypotonic environment, then we would see that that cell would burst, right? So that's something that is of consideration, is whether or not that cell wall helps to keep the cell from bursting in a hypotonic environment. And therefore, the reason that most bacteria enjoy a hypotonic environment, but some of them don't. Let's look at the opposite scenario, and I thought I would pick on Shannon just a little bit. She likes to hunt, and I think there probably are some others of you that maybe didn't mention that on your note card, but that also enjoy hunting. And I am curious if any of you have ever done any preserving of your of the leather from the animal that you've killed, and whether or not any of you have ever done any making of beef jerky and preserving the meat through the formation or the salt treatment of the meat. This is all about osmotic pressure. So the preserving of meat through curing with salt is because of the opposite environment than we looked at before. That is, putting a ton of salt around a cell creates a hypertonic environment. And in a hypertonic environment, the water is going to rush out of the cell, and the cytoplasmic membrane is going to shrivel, and we call that plasmolysis. So as the water rushes out, the cell membrane shrinks, it pulls away from the cell wall, and this is called plasmolysis. The rough thing about a hypertonic environment for a cell is that cells don't have any protection against plasmolysis. And in fact, in a very hypertonic environment, they die. This can be a really good thing, right? Because when we preserve meat using salt, that actually is a very effective way to kill bacteria. So salt-preserved meat, beef jerky, can sit out on the countertop for long periods of time, and it doesn't go bad. And that's because the bacterial cells that might have contaminated it have undergone plasmolysis, and they've been killed by that hypertonic environment. So if you would like to email me for extra credit, a little bit about experiences that you might have had with hypertonic environments and how they can preserve meat, or maybe you've been in a lot of you like to travel. I know, related to me, that passion for traveling. Maybe you've been to a place where the use of a hypertonic environment is commonly used in preservation. Tell me a little bit about that, and I would love to receive your emails. Do know, however, and you can email through Canvas, do know, however, that I am a bit behind on my emails, so it may be a while before I get you extra credit, but I promise that I will do so. I want to talk about a couple of lingo, sexy terms. These are protoplast and spheroplast. A protoplast is quite literally a cell without its wall. So if we have a cell that lacks, for example, if we've got a gram-positive cell and it completely lacks its thick peptidoglycan, we would call that a protoplast. That is, it would be nothing but the cytoplasmic membrane and no cell wall. A spheroplast is where the cell wall is partially degraded, though not entirely degraded. So coming back to our extra credit, what I want you guys to also tell me in your email, and both things for full credit, or for credit. So I also want you to tell me how we might make a spheroplast. That is, how might you take this gram-positive cell and weaken the cell wall, making a weakened cell wall or a spheroplast. That is what chemical that we talked about last time could be used to do that. Fantastic. We're now poised to move away from the cell wall and to layers that might be external to that cell wall. So hang on just a minute. I am totally going to grab a model and I'm going to show you where we're talking about when we talk about external to the cell wall. So recall our good old trusty model. We should probably name this guy. Oh, maybe you can include that in the extra credit as well, is a good idea for a name for our trusty bacterial cell model. But when we talk about the layers in the cell, remember that this yellow really does show the cytoplasmic membrane. So that's kind of trying to depict the cytoplasmic membrane. The green is trying to depict the cell wall. If this were a gram-negative, the green would be depicting the periplasmic space, the peptidoglycan layer within that, and the outer membrane. So it's not a very detailed model, right? We'd have to way zoom in on this to get any kind of detailed model, and we can do that, right? We can zoom in and then we get the other model that I've been showing. But I want to talk now about structures external to that. So some bacterial cells have another layer around them. In fact, we could actually say that they wear a jacket, they wear a coat, another protective layer. And these outer layers can take a couple of different forms. One of the most famous of these outer layers is called the glycocalyx. Glycocalyx is a broad term to say that there's generally a carbohydrate, a polysaccharide layer outside of the cell wall that is very, very protective. Glycocalyces, that is plural, can take more than one form. So they can be either a capsule or a slime layer. And in both cases, they're very protective. In lab, we're going to talk in detail about capsules and their nature. Slime layers are a little less distinct than a capsule, but in both cases, they provide a lot of protection. For example, if a cell has a slime layer or a capsule, specifically, really, if it has a well-formed capsule, it has a very good defense against phagocytosis. So picture this, if our bacterial cell here, I don't know what we're going to name it, Herbert or something, if our bacterial cell is trying to get away from an engulfing white blood cell, right? This white blood cell trying to come in and grab it. If it has an outer slick layer, capsule, it's going to slip away from that phagocytic white blood cell, right? So it's going to allow it to be a slippery little sucker and to get away from a phagocytosing white blood cell. Slime layers can have some of those same effects. It can also prevent hydrophobic toxins from getting in. Thomas will be interested to note that it can also protect from viruses binding, adhering, and being able to inject their nucleic acid. So there's a lot of protection afforded by capsules and slime layers. Another external layer that we sometimes see different than a glycocalyx is an S layer. The thing that makes an S layer different is twofold. One, it's the nature of the molecules making it up. That is, it tends to be protein rather than carb. So protein or glycoprotein, remember glycoproteins have carb regions on them, but we tend to see a polypeptide nature to an S layer. The other thing that differentiates an S layer is the composition, the organization. In an S layer, the glycoprotein or protein forms these very regularly repeating units, and it forms eventually what looks like floor tiles or ceiling tiles over the layer of the cell. So if you look really close at the ultrastructure, it'll look like this tiny little, very regularly repeating set of floor tiles. Sometimes they're even hexagonal. And what's cool about that is it's very, very regularly repeating. That leads to very succinct pore sizes in between the tiles in this outer protective layer. And so Ben and Sally are going to be super jazzed to know that this means that S layers can literally and have literally been used in biotechnology as membranes. Think of membranes with the smallest possible pore sizes and the most uniform possible pore sizes you can imagine. And in fact, if you go to the Web of Knowledge or the Web of Science, you're going to find a review article, a 2011 review article that's just entitled The Nanobiotechnology with S Layers. So S layers have a lot of application in biotechnology. But what do they do for the cell? Well, they're going to be very protective. They're going to protect against pH fluctuations, osmotic stress, enzymes. They're also going to protect against phagocytosis and toxins. So they have a lot of the same functions that a glycocalyx has. They also protect against predaceous bacteria. Later this semester, we're going to talk about a bacterium called Delovibrio. It just sounds scary, Delovibrio. Delovibrio is a predaceous bacterium that literally rams into bacterial cells. It actually moves them with its force, and then it starts rotating its flagellum, and it bores a hole into the outer membrane of gram-negative prey cells, and then it lodges itself in the periplasmic space, and it grows into a long worm-like structure and undergoes what's called multiple fission and breaks into many, many daughter cells, and it basically eats the prey cell alive. So that's what predaceous bacteria are all about. The other thing is that this may help the cell maintain its shape, rigidity, and promote adhesion. So we kind of recall that as being functions that we see of the cell wall, and so they're aided or aided and abetted by the S layer. Not all cells have an S layer. The most famous one that has an S layer is called Caulobacter crescens, and it's fun to look up and see some of the applications that has in biotechnology. Lastly, I want to talk about sheaths. Sheaths are another type of external structure, but rather than being a structure external to a single cell, they're a structure external to a population of cells. So we would have a whole bunch of cells housed by this outer sheath, and a sheath is kind of like a straw. You guys are totally going to laugh at me, but I wanted a demo, I wanted a model, and so a piece of one of the tubes that connects the gas to the outlet bit the dust, and I made a model. So you can see here, this is what a sheath would look like. So if each one of the beads inside of there are photosynthetic bacteria, such as cyanobacteria, and the outer tube is the sheath, see how this is enclosing a community. You've got inside of there enclosed a community of photosynthetic bacteria. And imagine how protective a sheath is to them. They've got this whole nice population cordoned away from predaceous bacteria and other threats. But also, this can hold on to surfaces. So, if my hand is a rock, here's this guy flapping in a stream or something like that. And hey, think about that. All the nutrients and food in the stream are flowing by this population of bacteria, and it's like fast food. They're like, woohoo, here comes my delivery food as it comes through on the water. So that's a pretty convenient thing. Let's summarize down in the picture below. Broadly, there are several layers external to the cell wall. One type are the glycocalyces that are polysaccharide, either slime layers, which are diffuse, or capsules, which are very, very discrete and regular. S layers made up of protein or glycoprotein, they're distinguished by their floor tile-like pattern. And finally, sheaths are a tube that holds a chain of cells together and gives them advantages in that chain.